Okay, so this worksheet is about one of your core biology practicals that you have to do, and it's how does light intensity affect the rate of photosynthesis. So we've got a bit of an introduction here. So we've got some microscopic algae. This is what it actually looks like if it's scooped out of a pond. You can kind of see a pond in the background, and that contains chloroplasts and is able to photosynthesize. And what scientists often do is they trap that algae inside little jelly balls. I'm not sure if you can see them here or here, and then they're referred to as algal balls. And what they do in this experiment is they put the algal balls into an indicator that changes colour as carbon dioxide levels change. We know that when carbon dioxide dissolves in water, it becomes slightly acidic. So under normal conditions, the indicator is a red colour, but this changes to yellow at higher carbon dioxide concentrations and purple at lower concentrations of carbon dioxide. So this is the method that you would do if you were doing this practical. So you'd pick out 10 to 15 of these balls, pop them into several different bottles, and then you'd add the same volume of indicator solution to each bottle and put the lids on. You would compare the colour in the tubes to the pH chart to work out the pH at the start. Then you set up a heat filter, which is a beaker of water, between the lamp and the tubes, and you cover one bottle in kitchen foil so that it's in the dark. So on the back, I've actually got a picture of that here. Okay, so there's the lamp. Here are our bottles, okay, with our algal balls. Here's our bottle wrapped in foil. Now the one thing they haven't added in here, which we would put in there, is a beaker with water inside it, and that acts as the heat filter. Okay, and basically any heat that is generated from the lamp, instead of passing to the bottles and warming up the bottles, and obviously the ones nearer the light would get hotter and these ones wouldn't, by having a beaker of water there, the light can still pass through, but the heat is absorbed by the water. So the temperature of this one and this one hopefully then would remain the same. So then it says, place the bottles at 10 centimetre distances from the lamp, put the bottle covered in foil next to the bottle closest to the lamp, turn on the lamp and leave it for 60 minutes. Then at the end, compare the colours of all the bottles with the pH chart, record the pHs of the solutions in your bottles in a suitable table. For each bottle, calculate the change in pH per hour. Add these calculations to your table. Okay, so if you imagine we've done this experiment, we've got our light, we've got our bottles with our indicator, we've got our algal balls in each one, and we've got our heat filter in between. So they're asking us, what is the independent variable? So that's the thing that I changed in my experiment. Well, if I look here, okay, what am I changing each time? I'm changing the distance from the lamp, but that's also the light intensity. Because I know, that if you were looking at the light from here, it would be darker than it would be from this point at the front. So what is the dependent variable, the thing that I'm measuring? Well, what I'm measuring is the change in pH. Okay, I'm not just measuring the pH, I'm doing a start pH and an end pH. So what I'm actually ending up with is the change in the pH. So name 3 factors that had to be controlled, kept the same, so that the results were valid and could be compared. So what if I had to keep the same? Well, let's look at this experiment here. Okay, I'm going to keep the same number of algal balls. I'm going to keep the same volume of indicator. Now it's really, really important that you write volume there. You can't put amount, they won't give you a mark for amount because we measure liquid in volume, so we have to say volume. So that's the same number of algal balls. The temperature, I'm going to keep the same, okay, and I'm going to do that by using my heat filter. I'm going to use, it says here actually, where did it say? To put them in a dark room. Have they said that? Nope, they haven't said that. So it's in the dark. Well, actually, what I want to do is I want to make sure I am going to do this experiment in the dark, because I want this to be the only light source in my experiment. You know, if this side's next to a window, they're going to get more light from the window, and that's not going to, I can't then compare this one to this one because they've had different amounts of light. So what I am going to keep the same as well is I'm going to keep background light the same, or I'd say, you know, make sure I do it in the dark. I'm going to keep the time the same. It says that on there. I'm going to do it for 60 minutes. There's several things there that I could have written that I need to keep the same so that I can compare my results. Okay, so why was the heat filter needed? Well, remember, anything to do with temperature, the first thing that should pop into your head are enzymes. Okay, so temperature is always about enzymes. We know that photosynthesis needs enzymes, and we also know that as temperature increases, enzyme rate increases, okay, until it gets too hot. And remember, when it's too hot, the enzymes are denatured, and then at that point there, the active site has changed shape so that the substrate's no longer fit, and then my reaction would stop. So I have to do this. I have to make sure that the temperature is always the same, because otherwise temperature itself will change the rate of my photosynthesis. Okay, let's have a look at the questions on the next page. So here, the bottle in foil is called the control. What is this for? So what the control does is it always removes the independent variable. Okay, so it removes the independent variable, and in this case the independent variable, remember, is the light. Okay, so basically I'm doing the experiment without the light, because what I'm looking for here is, would the liquid, would the indicator change colour anyway? Is the light causing it to change colour, or is something else going to cause it to change colour anyway? Okay, so what I'm looking for is the light causing the colour change, or is it something else? And they're always question usually comes up in the exam about control, so it's really important that you remember that the control is where we do the experiment exactly the same conditions, apart from we remove that independent variable. Okay, so these are our results. So we've got purple, pink, reds, orange, all the way up to yellow. Okay, so we know that something's happened inside these. Okay, so that something's happened with the carbon dioxide levels to cause them to change colour. Right, so it says now we've got our results. So here's our distance from the lamp. Here's our pH at the start, pH at the end. What's the change in pH? And therefore, what is our rate of photosynthesis, which is our change in pH per hour. So for each bottle, calculate the rate of photosynthesis as the change of pH per hour. So first of all, let's calculate our changes. So that one hasn't changed. That one's increased by 4, increased by 2, gone down by 1, down by 2, down by 2. So what is our rate? Okay, so the change per hour, and we know that we did this experiment for 60 minutes, which is an hour, so our rate is the change divided by 1, because we did it for 1, so 0 divided by 1 is 0, so 4, 2, minus 1, minus 2, minus 2. Okay, so now let's look at our results. So here they've given us our given us the equations to help us calculate it here, but we've done it already. So now we have to plot our results on the graph. Plot the variable you've changed, the independent variable on the x-axis, and the dependent variable on the y-axis. Okay, so let's get some graph paper. Okay, so here's my graph paper. So I'm going to draw my lines on the graph paper. Okay, there's my y-axis, and here is my x-axis. Now it says plot the variable you've changed on the x-axis. axes. So the thing that I've changed, okay, is going to be my distance, okay, because remember this is going to be my independent variable, and the thing that I measured is my change, or my rate here, which is my dependent variable, and that's going to go on the y-axis. So my value, my highest value is 50, so I need to go from 0 to 50 along my x-axis. So here 0 is going to be there, so I could go 10, 20, 30, 40, 50, but that'd be quite small, so I'm going to go 10, 20, 30, 40, 50. Right, I'm going to write here what that is. So that is my distance of bottle from lamp, and my units, centimetres. Okay, because if you forget to put your units, you're going to lose marks in your exam. So now I'm going to plot my rate. Okay, so my rate of photosynthesis. Okay, now my rate goes from 4 to -2. Okay, so that's why I've drawn my axes so that I can go into negative numbers if I need to, which I obviously do. Okay, so I need to go up to 4. So again, I could go 1, 2, 3, 4, but they'd be quite small. So I'm going to go up in 2s. So 1, 2, 3, 4. And then the same coming down. So -1, -2. I only needed to go to -2, but I'll go to that. And again, I need to put my labels on my axes. So rate of photosynthesis, which is my change in pH per hour. Okay, I can just take that directly from my results table. Okay, now I'm going to plot those results. Okay, so here if I look at my results table, at 10 centimetres I had +4. Okay, so 10 centimetres +4. I'm going to put a cross. 20 plus 2, 20 plus 2, 30 minus 1, 40 minus 2, 50 minus 2. 30 minus 1, 40 minus 2, 50 minus 2. Okay, so now I could draw a best fit, and it does look like it, or I could draw it with a ruler, but to be honest, this probably suits a best fit line. So when you do best fit lines, you just need to practice them a couple of times, okay, before you do them. Okay, so I've drawn on my best fit line. So now we're going to have a go at answering the questions. So question 8 says, describe the pattern shown on your graph. So when they're asking you to describe, they're basically saying you just need to write down exactly what you see. Say what you see. We don't need any science, we don't need any explanation of what's going on, just what the graph shows. So let's look at our graph and write our answer. So here, okay, what we can see is we always start with the x-axis, and we know that as the distance of the bottle from the lamp is increasing, so that's how we start, as the distance of the bottle from the lamp increases, what's going on? So we now need to read that from our y-axis. Okay, and what's going on is that the change in pH per hour, okay, so the rate of photosynthesis is decreasing. Okay, the change in pH per hour, or I could have written the rate of photosynthesis, is decreasing. Okay, but it doesn't just carry on decreasing. At the point here, it starts to level off. So I'm going to say that in my graph as well, in my answer as well, because at the moment it just looks like as the distance increases, this is going to continue to fall, but that's not the case. So from 40 centimetres on, the rate of photosynthesis remains the same. Okay, so now let's have a look at the next question. So now it says, explain why this pattern is observed. Remember that carbon dioxide is dissolved in water to make it acidic, which has a lower pH. You therefore need to think carefully about what's happening to the CO2 when you answer this question. Okay, so let's look back at our graph and think about what's going on. So this was the starting pH there, okay, and we know that from here, these 2 points here, these distances, there was an increase in the pH. So if the pH is increasing, it means it's becoming more alkaline. And if it's becoming more alkaline, okay, as we go upwards, it means that there's less CO2 in the liquid. And where has that CO2 gone? Well, obviously the plant has used it in photosynthesis. So this one here, the closest one at 10 centimetres, this one has increased the most in terms of its pH. It's become the most alkali, so therefore it's taken the most carbon dioxide out of the liquid and is increasing, its rate of photosynthesis is the most. Okay, at this end, however, we've got a fall in pH. Okay, so if something's falling in pH, it means it's becoming more acidic. So that means that we've got more CO2 being added to the liquid. So where's that coming from? Well, this is what lots of students forget, that actually that plants are living organisms, and because they are living, they are also doing respiration. And if they are respiring, remember, they're taking glucose plus oxygen and they are creating carbon dioxide plus water. So these plants here, okay, at these distances, let's look at our picture again here, these ones, they're probably still doing photosynthesis because they've still got a little bit of light, so they're taking some carbon dioxide out of the water, but they're also putting carbon dioxide back in because they are doing respiration. These ones will be too, they're also respiring, but they're photosynthesizing much more than they're respiring, whereas these are respiring more than they're photosynthesizing. Okay, so let's look at what we're going to write for our answer to that question. So the question said, explain why this pattern is observed. So we can say, so question 9, the closer to the lamp, the more CO2 is used as the liquid is becoming more alkaline. So this means that photosynthesis is occurring at a faster rate. Okay, and now we're going to put here, so at 40 and 50 centimetre distances, the plants are respiring more than they are photosynthesizing. So they are adding CO2 to the liquid and the pH is falling. Okay, right, now let's go on to the next question. So question 10, explain whether the pattern on your graph obeys the inverse square law. We can't do that, okay, because I took a bit of the information out from the questions earlier on, so we're not going to be able to do that question, so we're going to ignore that one. And then question 11, complete the missing words in the sentences below to explain the point of the tube covered in foil. So the part of the experiment in which the independent variable is not applied is called the control. The control is used to check that the independent variable has an effect and that the effect is not due to another variable. In this experiment, the independent variable is the light intensity. The control is the tube in foil. We know that the independent variable has a direct effect on the final pH of the indicator because the pH didn't change.